Hi, I'm David Benacom, and I'm going to present the results of our article, Motor Recovery through Perineuronal Net Modulation in a Parkinson's Disease Mouse Model, a study that was led by Camille Chatin and myself. We will start by introducing perineuronal nets, or PNNs. PNNs are a form of specialized extracellular matrix that forms around inhibitory parvalbumin cells during cortical maturation. PNN's deposit coincides with the onset of PV function and the stabilization of the excitatory-inhibitory balance. PNNs are also associated with a state of restricted plasticity, and PNN degradation in adults has been shown to enhance plasticity in sensory cortices. However, PNN in the motor cortex have been largely overlooked. In our first experiment, we transiently degraded PNN into the primary motor cortex using chondroitinase ABC. This treatment led to a temporary rotational behavior, suggesting an imbalance between the two motor cortices. This behavior is somewhat similar to that of hemiparkinsonian mice. Therefore, we next investigated primary motor cortex PNNs in the 6-OHDA mouse model of Parkinson's disease. In this model, a toxin is injected into the medial forebrain bundle, inducing the death of dopaminergic neuron in the substantia nigra and ventral tegmental area. This leads to a characteristic unilateral rotational behavior, along with a more general alteration in motor function, as measured here by the rotarod task, where mice must balance on a rotating rod. Following 6-OHDA injections, we measured PV and PNN numbers in the primary motor cortex. We found that PNNs around PV cells temporarily decreased at 2 weeks post-injection, returning to baseline level by 5 weeks. This PNN recovery coincided with the end of the animal's lethargic phase, the period of most severe motor impairment. Notably, motor stimulation after 5 weeks failed to improve motor function, suggesting the system had reached a stable impaired state. From these first two experiments, we developed a model in which PNN level link plasticity and stability in the motor cortex. PNN levels respond to either a midbrain lesion or a direct chondroitinase injection in the cortex. However, the functional outcome of this plasticity depends on what kind of input the cortex receives. We therefore hypothesized that after 6-OHDA induces a long-lasting detrimental effect on motor function, a subsequent injection of chondroitinase into the motor cortex, combined with a motor training protocol, could help mice recover from dopaminergic loss. We therefore designed the following experiment. We first lesioned the dopaminergic neurons, waited for PNN levels to return to baseline, then degraded PNNs again, this time while applying motor training. What we found was a sustained improvement in motor function, as measured not only by the rotarod, but also in two other behavioral tasks that are described in the paper, suggesting a partial rescue of motor performance. Importantly, this rescue occurred only when chondroitinase injection was paired with motor training. Neither treatment alone was sufficient. Finally, we assessed molecular changes using a deep learning-based in-house image analysis pipeline. We measured the number of PNNs around PV cells, as well as the excitatory-inhibitory balance in the primary motor cortex using VGLUT2 and VGAT staining. We found that more PNNs were deposited around PV cells after chondroitinase treatment and motor training, and this was associated with normalization of the IPSI contra VGLUT2 intensity, along with a trend toward increased VGAT. This change in synaptic marker was also associated with a clustering of microglial cells around PV cells. Thank you for your attention.